Okay, so welcome to your first taster class. This is the week 1 and first class to introduction to the biology syllabus. Before we begin with knowing more about the ASA level curriculum and everything else, I thought I'll share a quote with all of you. Take a moment and read it. And to get started, I also wanted to hear a little from you about something that you learned in biology, a term or a keyword or any concept which you found really funny or very difficult and it got stuck in your head. What was that? You can go ahead and share in the chat. What was something that you studied in biology previously that sounded funny to you or so difficult that you couldn't forget that term? Maybe it's not just particularly from biology, but anything in science. You could also feel free to unmute and share. Harshada, Namrata, Payal. Electrolysis, okay. Harshada, you found it funny or you found it difficult?
Actually difficult
Okay.
because when we hear that word, like it's so sounds so hard and heavy to hear itself. So
I get it. Namrata has shared a photosynthesis equation, very difficult to remember, okay. What about you, Payal? Thermoregulation. Wow, y'all have chosen really heavy terms. Okay, keep sharing though. If you think about anything else apart from what you've shared, you can drop the term in the chat. With the interest of time, we will now move on to what my strange word was. So what you're seeing on the screen is the axolotl larva. This is actually a larva of an amphibian water lizard. It's not exactly a lizard, but it's a salamander. And its name is ambystoma. So this is something that stuck in my head when I was studying biology, because I had never seen an animal like this. It lives in fresh water. Mostly it is found in South American coasts, and it has these feathery gills. So it can breathe underwater, and it can also stay outside water and breathe through its skin like a frog. So this was something that stuck to my mind for a very long time. Yeah. So our agenda today, yeah, it does look cute. And people do keep them in aquariums also in different countries. I just thought I'll show you what we are going to be overviewing in the first class. I'll give you a brief introduction of what Cambridge ASA level syllabus is in terms with respect to biology. Also, I'll show you the assessment patterns, that is how the tests look like, what are the total marks, how many papers, and what is the pathway that Avsara is registered to, like how do you navigate yourself through the ASA level curriculum and then complete it, in how many years will this happen. Then I'll just briefly take you through the names of the topics that you will study in AS level. And then I'll also take you through the outline of week 1. In the whole week 1, what are some things that we'll be studying in biology? So beginning with what is AS and A-level Cambridge International Examination. So we call this as CIAE. Okay. Your Cambridge is IGCSE 10th grade, and 10th grade is IGCSE, and your 11th and 12th grade is CIAE. CI stands for Cambridge International. AE is Advanced Level Examination. So AS is Advanced Subsidiary and A is Advanced. So this is equivalent to 11th grade, and this A level is equivalent to 12th grade. But as you know, in Apsara, we have extended our AS A-level program to 3 years. So you will be studying AS for the first year. In the second year, you will complete your AS level syllabus. And if there is time, we also start the A level in second year itself. If there is no time, then you do A level in the third year. But the plan is to finish AS level syllabus in 1.5 years and A level in the next 1.5 years. Finally, you give your exam after every syllabus completion. That is, once AS is completed, you give the AS. Then when A level is completed, you give the A level exam. You don't give everything together. Okay. So Cambridge International AS level is typically a 1-year program of study, while Cambridge International A level is typically, it takes 2 years. So if someone wants to get the A level certificate, they have to give the AS and A. But there are schools where the students choose not to do the A level, they just give the AS level. However, in Apsara, we encourage students to give both, because that is equivalent to our Indian education system's 11th and 12th grade. If you don't give the A level, then you will only be an 11th grade graduate as per Indian education system. But in Cambridge, they allow students to just give AS level. So assessments take place at the end of each program. Most subjects can be started as Cambridge International AS level and then extended to Cambridge International A level. So you must be getting an idea that typically this is a 2-year program, but in Apsara, we have extended it to allow students to actually absorb the curriculum and internalize and perform better. We have extended this 2-year program to 3-year program. Now, by the end of this taster week, what are some things that you will be learning? First of all, whatever the subject is, like you have attended the biology taster, maybe after this you'll attend the chemistry or physics class. So the main aim of these tasters is to get an overview of whichever subject you are attending, and it will be the same. The Cambridge curriculum pattern is similar for physics, chemistry, and biology. You will also understand the structure of the assessments. Assessment means what the Cambridge board is going to test you for. What are they going to test in each candidate? Know how to show your skills to the best of your ability. Means what are some skills that is encouraged so that you can be more successful in AS and A level, and how will your study habits, support from school, learning resources, how will you use all of this to enhance those skills? You will also understand how this AS A level curriculum is a very reflective approach. Means you can track your own progress and you're encouraged to do more self-learning and have more self-awareness, because everyone chooses subjects of their own choice. So when you're choosing subjects, over there, your role has a higher weightage as compared to what is being taught. Because once you choose a subject, it also tells a lot about what is your strength, what is your weakness, what areas are you willing to look for in universities based on subjects that you take in AS and A level. So a lot of reflection is required in your 11th and 12th grade curriculum. You'll also learn how we can inculcate those skills of effectively reflecting on your own learning. Coming to how will you be assessed? How will Cambridge test you? So in AS level, you will be entered for 3 examination papers. So Ms. Tanuja will be registering you for 3 subjects papers. Not 3 subjects, whichever subject you are registered for, in that subject you have to give 3 exams. For example, Ms. Tanuja has registered you as a biology student. So you will have to give 3 exams in biology. And those are paper 1, paper 2, and paper 3. This is only for AS level. Paper 1 will be a multiple choice paper, but this multiple choice question will come from the theory that you're learning. Theory means all the chapters. Okay. Paper 2 pattern will be structured, very much like what you have experienced in your IGCSE level. Multiple choice paper and a structured paper. Structured paper means where you will have questions and you'll have to write sentence-based answers, subjective answers. Sometimes it will be a 1 mark question, sometimes it will be 2 or 3 mark or 4 mark question. Paper 3 is an advanced practical skill paper. Now, unlike your IGCSE level where you give alternatives to practical, where questions are very practical based and you have to write the answer, unlike that, this paper 3 has assessment questions where you have to do investigations. You have to carry out experiments live in the examination lab hall. Based on your experiment, you will get results. You will record the results on the answer booklet, and you will also give your conclusions, do some calculation. Apart from the experiment, there will be experiment related questions and more analysis, interpretation questions. That is your paper 3. In the course of this taster week, at the beginning of every class, I will try to show you some examples of different questions from paper 1, paper 2, and paper 3, so that you get the exam style idea, like how these questions look like. Now coming to A level, this is particularly your grade 13 in Apsara. In A level, you will be entered for 2 more exams, 2 papers. That is your paper 4 and paper 5. So all total in biology, you will be giving 5 papers, 3 in AS level and 2 in A level. Your paper 4 will be a structured paper again, but paper 5, instead of doing the investigation live, this time your paper 5 will be a planning, analysis, and evaluation paper. Means in this paper, you will be asked questions just like alternatives to practical paper in 10th. Have you all given the alternatives to practical paper this time in IGCSE? No, right?
No.
No. You had only given the MCQ and theory paper. But have you seen an example? Have you seen a sample paper? How the alternatives to practical paper looks like?
No.
Okay. So sometime in the upcoming classes, I will show you how these questions are set, where a student will be given a scenario and then you will be asked to plan an experiment and find out something. So if you want to find out, let's say, how fast or how slow photosynthesis is happening throughout the day, in the daytime versus when there is no daylight. So how will you carry out that investigation? What equipments will you need? What will be your method? What will be the aim of your experiment? What are the different dependent, independent variables? So all that will be asked in paper 5. So this is how your assessments are going to look like. Coming to the paper. How does paper 1 look like? Paper 2, paper 3, and so on. So right now on the screen, you're looking at paper 1, 2, 3 detail. Quickly, can you tell me in which year will you be giving paper 1, 2, 3 exam if you take biology? Is it AS level year or A level? Yes, correct. It is the AS level year. So I have one more checkpoint question. Suppose you start learning biology from this year. Which year will you be sitting for the exam? The biology paper exam. You have 3 years of AS and A level. In which year will you sit for the AS level exam? Any idea?
Is it 2023?
After
2023?
Will
1.5 years.
Yes. So you will not be sitting in 2023, but you will sit for 2024. Okay. 2024 Feb March. That is when you will sit for your AS level exam. And then, so the current batch that gave AS level now, they will give A level next year. And the current batch, which is now going to grade 12 now, after you, like just before you, they will give their AS level exam in the coming year, in the Feb March of 2023. So all of you are going to actually get 2 years to complete your AS and 1 year to complete your preparation for A level. So if you enroll for AS level in Avsara, your AS level exams will be on 2024. Paper 1 will be a 40-mark paper. Paper 2 is a 60-mark paper. And paper 3 is a 40-mark paper again. Again, quick recall, paper 1 is a multiple choice paper. 40 multiple choice questions will be asked. And you will answer, you choose one answer out of the 4 options. Basic, how your multiple choice papers look like. Then paper 2 is a structured paper. So you will have to write answers in the given space. These questions will be based on your syllabus, and this will also be 1 hour, 15 minutes. Okay, notice same time for paper 1 and paper 2, same duration, but paper 2 is 60 marks. Paper 3 will be for practical, the live practical exam, that is 2 hours. That will also have 2 very long questions. First question will be a wet lab where you are actually dealing with chemicals. And second question in biology is most of the time it's based on microscope study. So yes, that is how the papers look like, how long they take and how many marks they carry. In A level, paper 4 will be 100 marks for 2 hours. And you will have a total of 10 structured questions. This pattern has slightly changed. From 2022, paper 4 structure has slightly changed. So here it is saying 85 marks plus 15, but now it is no more like that. It is completely 100-mark paper with no sections. And paper 5 is a 30-mark paper, which looks like an alternatives to practical paper. You will not do the practical, but investigation and experiment related questions will be coming in this paper from your A level syllabus. I'll just pause over here for a minute so that in case you have a question, you can ask me in the chat. Is there anything that you would like to ask? Yes, Harshada, go on.
Miss Amrita, so in paper 4, we will give the paper for 2 hours, right? So how many questions would we attempt?
10.
10.
There'll be 10 structured questions. So it will be a very bulky paper, but you will only get 2 hours for that.
Okay.
Okay, then the next thing that I wanted to share is the pathway. How are we going to give the exam? So actually, Cambridge has 3 pathways. That is, it allows candidates to take only AS level. So let's say you take only AS level. and you don't take A level. So you will complete your AS level in 2 years, finish the exam, and then you graduate out of Avsara. But that's not how we register our students. We make our students sit for both AS and A. Okay, because in the future, if you choose an Indian college or university to study your bachelor's, then you will need both your 11th and 12th degree. So AS level is equivalent to the degree of 11th grade, and A level is equivalent to your 12th board. Here you can see what are the papers you give in each level. In AS level, you sit for paper 1, 2, 3. And in A level, you will sit for paper 4 and 5. Now, which pathway does our students apply for? Is it they only sit for 3 papers in AS level and graduate out of Avsara? Or do they sit for all the papers after 3 years? Means in their A level year, they complete everything, AS exams as well as A exam. Or is it this second pathway? They give their AS level papers in the 3 papers after AS and 2 papers after A. What do you think? Out of these 3 routes?
The second pathway.
Yes. So in Avsara, we prefer to register our students in the second pathway, where students give their AS level papers, finish them off, so that one burden is off your shoulder, and then you study for A level and give the 2 A level papers. Okay. All right. Next is about how do you get your grade. So what will be the final grade after you complete your AS and A level? So first, when you give your AS level exam, you will receive a grade, but that is not your final grade. 50% of your AS level result, that is, suppose you have scored, so your total AS level marks is 260. Okay. So out of, no, not 260, it is 60, 40 and 30, 130, sorry. Out of 130, suppose you have scored 100. Scoring 100 out of 130 will give you a temporary grade. That is your AS level marks and AS level grade. Then 50% of that result will get added with your final A level grade. So weightage, you will learn about weightage later. When we come close to exam, I'll tell you how many marks, how much percentage comes from which paper. It will be too confusing for now. But one thing that you need to know is that final grade that you get after your A level is not just your A level marks. So for example, let me show you here. Let's say you have scored 80% in your AS level. Okay. And then you have scored 75% in your A level. But your final grade will be taken from both AS and A level. 50% of marks from your AS level and 50% of your marks from A level will be added together and a grade will be given. So what is 50% of 80%? You scored 80% in AS. So how much will Cambridge take from here? Any thoughts? Share in
40%.
40%. And in A level, only in paper 4 and 5, you have scored 75%. What is the 50% of this? 75 is half. That will be
37.5.
Yeah. So 37 plus 40 is how much? 77%. So that will be your total percentage. And based on that 77% marks, you will be given a grade. So what does this mean? It means that your AS level results are very important. They play a very important role in giving you the final grade. Because in AS level, you sit for 3 papers, 130 marks. In A level also you sit for 2 papers and those are also 130 marks. Equal weightage. So 50% marks comes from your AS, 50% comes from A, and then a final grade is given to you. So Namrata, it is like you give your 11th grade exam, you give your 12th grade exam. Finally, in the certificate, the grade is based on your performance in 11th and 12th together, not just 12th. So to be able to succeed in 12th, you need to be strong with your concepts and whatever is taught in 11th grade. So if you have struggled in 11th grade, it will affect your final result. And this is the same case for all the ASA level subjects. Okay. So that's how your marks will be. So currently the students who just gave their AS level exam, they have not yet, they will not receive a final grade. They will receive a temporary grade. That is only their AS level grade. Once they give their A level and AS level marks will be mixed and a final grade will be given. 50% from each will be taken and a final grade will be given. Okay. Okay. We will now just dive into the topics that we are going to study in biology. So we name these topics based on units. There is a total of 11 units in AS level. Your first unit is about cells. So you have studied about cells in IGCSE, and now you're going to study about the details of cell structure and functions of those structure in AS level. So definitely you will learn about many more structures inside the cell, which you haven't studied in 10th grade. So there'll be a detailed understanding of cellular structure. You will also learn about microscopes and how to use microscopes to study cells. In unit 2, you're going to learn about biomolecules. Again, you had studied about biomolecules in 10th grade, but very briefly they were introduced to you. Can you recall what were some things that you learned about biomolecules in 10th grade? Already the names of the biomolecules are here. Which ones have you studied and which ones you have not? Do you recall anything? Hi, Anam and Misbah. You all joined later, so I didn't get a chance to greet you. Yes, Namrata, go ahead.
I just remember we not talked about very detailed, but we learned the enzymes which break down these molecules.
You
Protease,
learned about
lipids.
yes, you learned about amylase. Amylase will digest starch. Starch is a carbohydrate, isn't it? Then you studied about lipases, which break down fats and lipids, and proteases, which digest proteins. So that you studied in enzymes unit. But before you learn about enzymes, you need to know what are these big biological molecules that make up our cells. So actually, we say that cell is the basic unit of life. But what is a cell made of? A cell is made up of these things. And you're going to learn that in the second unit. And thanks for mentioning, Namrata. Our third unit is again all about enzymes. But as I told you, whatever you are learning in your IGCSE level, you're going to build and learn more complex details about those topics in ASA level. So here in ASA level, you will learn a lot more about the chemical structure of enzymes and what makes an enzyme work faster or what makes an enzyme become slower or ineffective. Okay, so you're going to learn a lot more about the biochemistry of enzymes. In unit 4, you're going to dive deeper into just the structure of cell membrane. So unit 1, you learn everything about all the parts of the cells, but in unit 4, you only zoom in and study the cell surface membrane and all the membranes that are there in your cell. What do they do? What is their role? How do they help in transporting things into and out of the cell? How do they send signals? How can one cell send signal to another cell? All that is taught in Unit 4. Then comes Unit 5 and Unit 6. In Unit 5, you're going to learn a completely new concept, that is division of cell, how cells divide. So there are two main ways in which cells divide, but you are only going to study about one, that is mitosis. So when a cell divides, it has to follow a very disciplined path, and that is called the cell cycle. You're going to be learning about that cell cycle. Cell cycle tells you about the stages in which a cell grows and then divides. So that is Unit 5. It's called cell cycle and mitosis, or mitotic cell cycle. In Unit 6, you are going to learn about a new biological molecule in a lot more detail, that is nucleic acids. Your DNA, your RNA, that make up the nucleus of the cell. What is their function? What is the structure? How these DNA and RNA help in making proteins, and why proteins are needed by your cells. All of that will be taught in Unit 6, nucleic acids and protein synthesis. Synthesis means formation, making something newly is synthesis. Next comes Unit 7 and Unit 8. Unit 7 and Unit 8 is more about systems, organ systems in living organisms. So far, till Unit 5 and 6, you were only learning about cells and different molecules that make up the cell. What is their function? In Unit 7 and 8, you will learn about the transport system, that is xylem and phloem in plants, how they help in transporting water and food across the entire structure of a plant. You're also going to learn about transpiration. You will learn about factors which increase transport or slow down the transport, the adaptations that xylem and phloem have. You are also going to see sections of stem, root, and leaf under the microscope and actually see the xylem and phloem cells. How do they appear? And based on their appearance, you're going to justify how they are being able to do their function. How is the structure related to function? Similarly, so this is the transport system in plants. You will also study about transport system in animals. Guess what you will be studying in Unit 8?
Circulatory system.
Circulatory system. You will study about the structure of heart. You will study about the cardiac cycle. That is, right now you're looking at a beating heart. So every time a heart beats, there is a systematic way in which your heart will beat. The different chambers of the heart rhythmically contract and relax. Why is that happening? And what will happen if that doesn't happen rhythmically? How will a person maintain the health of their heart? So knowledge of all of this helps us in treating many cardiovascular diseases. Not only heart, you will also learn about arteries, veins, capillaries. You will learn about hemoglobin, the protein which captures oxygen in blood. You'll learn a lot about blood and blood cells and hemoglobin in this unit. Again, Unit 9 has another organ system, that is gas exchange. And that relates to the structure of your respiratory system and how the health of the respiratory system is affected due to tobacco smoking. Unit 10 is about infectious diseases. What happens when foreign pathogens or other organisms try to invade your body? So there are many types of diseases. Some diseases are not passed from one person to another, but in your syllabus you have infectious diseases. That means these are diseases which are communicable. If one person or one animal or one plant gets the disease, whoever it is coming in contact with or sharing the atmosphere with, they can also get this disease. So it transfers from organism to organism with the help of a pathogen or a disease-causing organism. The last unit in your AS level is immunity. Here you will learn about what is antigen, what is antibody, how do white blood cells in your blood defend your body from outsiders or organisms which are not a part of your body. How is your body protected from that? You're going to learn about B cells, T cells, B lymphocytes and T lymphocytes. You will learn about vaccines. What is the importance of vaccine? You will learn about WHO, the World Health Organization, and how does it contribute to spreading awareness and treating epidemics, pandemics in different parts of the world and raising awareness. So all that will come under Unit 10 and 11. Unit 10 and 11 is all about health, diseases, immune system, vaccines, antibiotics, okay? That's all. That is all about all the topics in biology. So it is 9:05 now. I would like to show you the outline of this week. What are some things that we will be doing in this whole week? So today is Tuesday. As you can see in the taster week 1, we are getting just a small sneak peek. I won't be able to teach you the whole unit because we have only 5 days. So in this taster week, we have 4 days of learning: Tuesday, Wednesday, Thursday, and Friday. And the 5th day is a quiz. So in this whole week, you will get an idea of how is the study life of an AS biology learner. How much do they have to study in the class? What are some preparations that they have to do after the class? Like you can see over here, after class, mostly we have post reading or worksheets to solve. Sometimes before a class, I'll give you a pre-reading. Maybe instead of post reading, I'll give you a pre-reading that is going to be helpful in the next day's topic. Okay, so things like that. And then finally on Monday, we'll have a short quiz based on what we studied this week. And I'll give you some feedback on how to do well, or how does my feedback look like on the test paper, or how do we do feedback? You'll get an idea. Now, the problem is when we learn anything online, we post our tasks and activities on Edmodo, isn't it? But because in the taster weeks, different students will come for different subjects, we can't have an Edmodo class for that. So what we decided is tomorrow in the taster class, I will be sharing a link to a Google Drive. The name of the Google Drive would be Biology Taster 2022. And in that drive, I will be dropping all your reading homeworks, worksheets, so that you can solve them and you can also put them or submit your work in the drive itself. Okay. So in the drive, you can create your own folder with your name, and I will, whatever homework I give, you solve it and you drop it in that folder. Okay. We'll talk more about this Google Drive thing in tomorrow's class. Do you have any questions regarding the outline of the week? Or do you want to ask anything about what you need to be ready with? No questions? Okay, thank you, Namrata. So what we are going to learn now is going to, or whatever activity we are going to start with today, it's going to be testing your prior knowledge, means your previous knowledge from what you have studied in IGCSE coordinated science in your 10th grade. So this is like a transition path. You are learning the same topic, you're learning about the same things, but right now you are going to learn a little more detail, or you will see the complexity of that same topic. Simpler introduction facts were taught to you in 10th grade, but now that you're moving to ASA level, that same topic will be there, but you will be learning more details about that topic. So you are on this path of transition right now. So in a prior knowledge activity, right now you need a blank sheet of paper and a pen. Just give me a thumbs up if you have all of this with you. If you don't have, quickly grab a notebook and a pen, or pencil would be better because we'll be drawing something. Do you have pencils with you? Thank you, Harshad and Namrita. Payal, Misbah, Anam. Okay. Anam, do you have a pencil and sheet? Okay, great. So what will we do now is we are going to draw something. So since we are learning about cells, the first thing I would like you to do is recall whatever you have learned about plant and animal cell in 10th grade and just draw a simple animal cell and plant cell diagram. Draw whatever parts you remember from 10th grade and just label them. Once you finish drawing, add your labels. If you want to annotate, annotate whatever you want to. And then you can tell me that I'm done. So I'll ask you to display your diagram on the camera. I want to take a look at your diagrams. Okay, let's take 2 minutes. Make our diagrams of animal and plant cell, whatever we remember from grade 10. Label all the parts. You can reply as done in the chat if you're done with the diagram. Let's take another few seconds to finish our diagrams. plant cell and an animal cell. Okay, please give me a heads up if you are done, or how much time will you need. Please let me know in the chat. Okay. We just need to have one basic diagram. It doesn't have to be very detailed and artistic. Thank you, Misbah. Misbah, whenever you are ready, you can show me your diagram on camera. If anyone else is also done, let me know. I'll be looking at the camera panel then for your diagram. Okay, I can see your diagram, Misbah. Can you just pan your notebook a little closer so I can see the plant cell first? If you can just show me one cell at a time, it would be easier. Wow, I can see label lines. There is animal cell with cell membrane, cytoplasm, and nucleus. Okay, I can see some dots have been drawn in the cytoplasm. Okay, now you can show me the plant cell. Plant cell has nucleus. Oh, where is the nucleus of the plant cell? There is vacuole, cell wall, chloroplast, cell membrane, and nucleus is missing, Misbah.
Yeah,
Isn't it?
yeah, it is missing.
We can add it.
Yeah.
Okay. Thank you for sharing, Misbah. Both diagrams are correct. Let's see if anyone has any more labels to add. Go ahead, Namrata, Payal. You can show me your diagrams. Payal, is it possible to sit somewhere where there is light? Because it's too dark where you're sitting. Will you be able to go near the window or something? No?
Yeah, just give me a minute.
Can you switch on the light?
Okay, yeah.
Namrata, you can set up your camera meanwhile. Anam, Harshada, are you guys done?
Yes.
Okay. Payal, I'm trying to, but it's not visible. Okay, plant cell. You can see the vacuole, nucleus, cell wall, cell membrane, cytoplasm. Awesome, great. Is there a chloroplast also? Yeah, I can see. Yes. And where is your animal cell file? Okay, here it is. Okay, I can see. Thank you for sharing. Namrata? Namrata, where is your plant cell nucleus? Okay, that is the nucleus. And those tiny dots are chloroplast. Okay. Okay. And animal cell? Animal cell nucleus is not there and there is a vacuole. How will the animal cell survive without a nucleus? Nucleus has to be there, no?
I could remember much about the animal cell.
Okay, you can rectify it now that you know. Thank you for sharing. Harshada, I can see. Can you show me one cell at a time? Show me the plant cell first. Okay, okay. Yes. Harshada, that is not chlorophyll. That will be chloroplast. Inside the chloroplast, there is chlorophyll. So that's okay. Show me the animal cell. Yes, yes. Is that the nucleus or what have you written? Yes, correct, correct. Okay. Anam, do you want to show your diagram? Hi, Anam. Anam is hiding.
Can you see?
Yes, I can see. Show me one cell at a time so that it's bigger. I just bring it a little closer. Yes, yes. Okay, there's a large permanent vacuole. There is a nucleus, lots of chloroplast, cytoplasm. Awesome. Show me the animal cell now. Okay. What are those black dots in the cytoplasm? Small vacuoles.
Yeah, I read this.
Okay. Yes. Yes, correct. Correct. Thank you for sharing, everyone. I was just thinking, all those labels that you have made, do you know the function of those parts? For example, Anam has labeled small vacuoles. Harshada and all of you have shown chloroplasts. Do you know the function of these structures that you have labeled? If you know, then just share one of those structure and its function. When I'm saying structure, I'm talking about the things that are there inside the cell. Go ahead and pick any one structure and write its function in the chat. So name of the structure and the function. Any one. Each one of you can share one. So you can write about nucleus, about chloroplast function, or if you want, you can write down the function of cell membrane, cell wall. Vacuole will store starch. Vacuole actually stores water, sugar, salts, minerals, not starch. Glucose is correct, but mainly, Namrata, vacuole looks like a water balloon. It's a main water storage part. So water is there. Do you remember this word? All of you? Sap. Do you remember what sap is? So sap is the watery liquid that is found in the vacuole. So on the screen, you are now looking at a typical animal cell. Most of your diagrams had these parts, isn't it? You had the nucleus. The nucleus contains DNA. What is the function of DNA? It carries all the genetic material that we inherited from our parents. Yes, I can see more functions. Nucleus controls all the other activities of the cell. Chloroplast contains chlorophyll. And because of chlorophyll, photosynthesis can be done by plant cells. Thank you for sharing, Anam. Cytoplasm is a jelly-like liquid inside which all the chemical reactions of the cell occurs. Correct. Cell wall protects, and it is only found in plant cell, isn't it, Misbah? Protects the cell, supports it. Also, it gives shape, gives a definite shape to the plant cell. Cell wall is very rigid. Correct. Okay. So this is a typical animal cell. You can see the parts that you had labeled with their function. And this is a plant cell. This is a palisade cell. Palisade cells are found just under the epidermal cells of the leaves. These are found in the leaves. You can see that in the plant cell, the unique thing is the vacuole is so big that it takes up almost the entire area of the cell, and it pushes the nucleus and chloroplast to the sides. So you have, when you're drawing a plant cell, make sure the vacuole is in the center and it is pushing all the other structures to the side. So there is vacuole, there is nucleus, there is cytoplasm, but all of this is pushed to the periphery. Periphery means to the edge. Then there is a cell membrane. Two boundaries are there in a plant cell, the cell membrane and a cell wall. Cell wall is made up of cellulose and cell membrane is made up of lipids and proteins, which you will study in Unit 2, Biological Molecules. Take a moment to look at the labels, and under the label, I have written the functions. So chloroplast, look at the function of chloroplast. It contains the green pigment chlorophyll. And what does chlorophyll do? It traps sunlight for photosynthesis. Look at the vacuole. It contains sap. What is the meaning of sap? Sap is a mixture of water, sugar, salts, iron, all that is necessary to maintain water pressure, or because of that pressure, the plant cell has a turgid or a swollen like shape. And then finally, cellulose cell wall. Cellulose cell wall may be rigid, hard, gives definite shape to the cell, but one thing about cellulose is it allows water to pass through it so that the plant cell gets a supply of water. So it gives structural support. Okay. With that, I would like to end our lesson over here. Time is up. And the last thing I wanted to show you is an electron microscope. We are going to study more about this microscope in class tomorrow, because we'll see how our cells look like when we see it under this microscope. That's all for today. Thank you for attending the taster class, and see you tomorrow. We'll continue to learn about cells and microscope. Thank you, everyone.
Bye, Miss Amrita. Thank you.
Bye. Bye-bye.